All right. Well, welcome everybody to Under the Radar, Discovering a World of Overlooked Biodiversity with Tracy Feldman. Tracy, thank you so much for being here.
Well,
Many
thank you
species,
very much.
yeah,
Oh,
great. I'm just going to give a brief understanding of what we're all here to see, which is about many species living all around us are either poorly understood or have not been documented at all. Yet sometimes new species may be living right under our feet, waiting to be discovered. Tracy's going to give us updates and insights from 10 years of collaborative research about tiny insects that spend their young lives burrowing inside leaves and stems. He's going to talk about discoveries and new species as well as mysteries that are still unsolved. So this sounds really cool. And Tracy, welcome. Anything you'd like to say before we share our screen?
no, but thanks everyone for coming, and thanks for giving me the opportunity to do this.
Awesome. Well, we really appreciate it. So I'm going to share this and I'm going to open it. Oh, I'm going to move this to this
Okay.
side. I'm sorry, Tracy. I'm trying to
Hey,
move the.
Tracy, I'm going to interrupt real quick. Tracy, you want to explain your role with the city of Raleigh and what you do?
Uh-huh. Sure. I am an assistant manager at Durant Nature Preserve and Horseshoe Farm Nature Preserve, and I run environmental education programming, but I also do this work. And some of this work was done through my work with the city, but most of it was not. So this proceeds and is happening simultaneously along with the work at the city. So my work at the city touches on this, but it's not. But this work is beyond what I do at the city.
Cool.
Thank you.
Sure. All right. So should I start?
Go for it, sir.
Okay. Awesome. So I want to start with a quote from E.O. Wilson. A lifetime can be spent in a Magellanic voyage around the trunk of a single tree. And I feel like that characterizes well the sort of types of journeys that I take. People who hike with me and want some hiking usually hate walking with me because I'm stopping all the time to look at small things. Okay, so next.
Well, for some reason, it is not advancing.
Got it. Okay.
Let me.
Yeah, and I don't have control, so I'll be saying
Oh, you
next a
know
lot.
what? Hold on. Give control. Tracy, try
Oh,
this real quick.
okay. I'd love to get control. Let's see if I can. Can you
Are
do it?
you are you able to advance?
I don't know. Um, I don't know where to advance it. I don't see anything to advance it
All
with.
right. I'm going to stop sharing and we're going to try doing this way then. And I am terribly sorry, everybody. We thought we had this all under control. So let's try this.
Okay. All right. Oh, great. So I want to talk about why we should catalog biodiversity in the first place and why North Carolina in particular might be an interesting place to do it. Next. Oh, oh.
Yeah, I'm
Is this
sorry.
going to be,
There we go.
can I do it? Or do you?
Here, let's see if
Um,
I'll try sharing control again.
okay.
Give control. There you go,
Okay.
sir.
I don't know how to do it though. I'm clicking with my mouse, I'm clicking with my down arrow, and I'm not seeing anything new.
All right. It's just going to be me then.
Okay, so next. Can you do it?
You see it?
No, I don't see anything new.
What do you see right now, sir?
I see the first bullet point.
Do you see why should we catalog biodiversity?
Yep. I'm asking for the next one. Yep. Yep.
And now do you see the next one?
Nope.
Interesting. I'm not sure. It's going forward on mine. Let me see. Resume slideshow. Ah, there we go.
So then I'll talk about leaf miners in particular next. And then some discoveries three types of discoveries. So first of all, new locations for miners that are known, new information about species we know, so maybe their behavior or new food plants that they might eat, and then entirely new species. Next. And then I want to talk a little bit about how we name new species, and next, and what to look for and how to find leaf mining insects, and some new discoveries, some new mysteries as well. All right, so I'll open it up to the group, I guess. Uh, why should we catalog biodiversity? Why does it matter if we know what other species are around us? Anyone have any ideas on that?
I have a lot of ideas about it.
Yeah, go for it. Yeah.
Because obviously, due to my role, um, I'm very concerned about preserving as much as is possible native biodiversity, um, to maintain the, I guess, the relationships that plants,
Mhm.
animals, etc., have
Mhm.
developed with each other.
Mhm. Yeah, great. So they have relationships, and these relationships matter. And if they change, if a species were missing or whatever, if it weren't there, or if there were too many of one species or another, then it might change how those relationships and might change how the ecosystem worked, how the community worked, functioned together. Sure. And in fact, we are also dependent on many species, many species. Sometimes we are dependent on those species directly, sometimes indirectly, and it's really important. So great. Next. And a lot of these, so we're also losing a lot of species. So extinctions are going up with human population. Next. So some people, an author, Elizabeth Colbert, has called it the sixth extinction. It's really the seventh, I guess. There was another mass extinction that she hadn't accounted for, but we're in the seventh mass extinction on Earth where lots of species are going extinct. But this is because of us. All right, so next. And in 2019, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services put out a document suggesting that over 1 million species are under threat of extinction now. And so why here? Well, we live in a very cool place. There are 36 places that are called biodiversity hotspots on Earth. They're really diverse, meaning that they have more species than other places around them. And also, or they have more species that are found nowhere else than other places around them. And if you look at if you go back, please, if you look at the southeastern United States, you can see that we are one of those 36 biodiversity hotspots. So right here, right near where we live, is very rich in biodiversity and rich in species that are found nowhere else. Next. But these places are in peril. So these biodiversity hotspots are actually being lost. We have only about 5 to 25% of each of these hotspots actually exists anymore. All right, next. And North Carolina is growing really fast. It's got the 10th largest growth rate in the country, and between 1 and 1.5% yearly. That means more land conversion and less habitat available. So we want to preserve as much habitat as possible. But if we preserve habitat, is that enough? Like, do we need to worry about the species? If we preserve habitat, have we got the species automatically? What do you think? Anyone want to weigh in on that?
Absolutely not.
I mean, we do need to preserve habitats, right? That is true, right? But at the same time, yeah, if we preserve them, we can't guarantee that the species we want to find are in them unless we actually catalog the species and know what's there. So go ahead next. Another way to word that question is: are apparently intact ecosystem or systems really intact? Maybe not. Next. So 3 questions you might be able to answer only if you know what species are there. Are some species missing or declining? Are some species becoming more common, maybe than they used to be? Or are they changing when they come out in the year? That may be important as climate change proceeds. Next. So I want to build an argument here that, so this is a one example of the showing the diversity of the Southeast. And so there's um, the plants are more diverse where it's darker blue and darker green, and so you can see that they're more diverse. Plant family diversity is great in is greatest in the southeastern United States. So um, I want to build an argument for why that might also lead to greater insect diversity. Now, insect diversity hasn't been assessed as much as plant diversity in some respects, but maybe butterfly diversity has and things like that. Few groups, but not all groups. All right, next. If you look at the biodiversity of plants and animals anyway, this disregards some biodiversity, but at least biodiversity of plants and animals, you can see that over half the biodiversity is insects. And next. And it turns out that half of that diversity, more than half of that diversity, are plant feeding insects. So there are huge numbers of insects that feed on plants. In fact, more than there are plant species themselves. Next. So to illustrate that point, I can show you a plant. So here's a plant and my great artistry. And next, they're fed on by a bunch of things. And so you might have things feeding on the leaf surface, like leaf miners, gall and leaf miners and gall insects living inside the plant as well. So you'd have things on the surface and things under the surface. I'll talk about more about leaf miners later and explain what they are. There are things that feed on the sap. There are things that next that feed on the flowers, that eat the petals, etc. Next, next. There are root feeders. There are pollinators that eat nectar and pollen. There are seed dispersers that may, and fruit feeders and seed feeders. There are protective ants that help sap feeders. There are mycorrhizal fungi that help get nutrients to plants. This plant is interacting with a large community of other organisms, just this one species of plant. And those things have predators and parasites. And those other things have predators and parasites. Next. And the predators and parasites have predators and parasites. Next. And there are decomposers that decompose them all when they die. So we can see, and each plant has its own chemical defenses that keep certain things from eating it and allows other things to eat it if they can get around those defenses, which means that if you have a plant next door, a whole other suite of insects could be feeding on that other plant that's a different species. So this can lead to large amounts of biodiversity if you have large numbers of plants, and we do. I'm going to speak in particular about leaf miners because that's work that I've been doing over the last 10 years. All right. So next. Before I talk about leaf miners and introduce those, I want to talk about the insect life cycle because that will factor into understanding leaf miners and how they work. So because it's a cycle, you could start anywhere, but we'll start with eggs. And by the way, if people have questions as we go, feel free to ask if over the chat, or let me know if you have questions and you want me to stop or like address those. All right, so we can start with eggs. So these things have eggs, and then all leaf mining insects are go into the larval stage from the eggs. The larvae hatch out of the eggs, and there may be multiple larval stages, and then they go into the pupal stage. And in the pupal stage, what they're doing is they're changing. They are losing things that they needed as a larva that they don't need anymore, and they're like extra legs maybe, or they're gaining things. I'll explain. I'll explain that soon, Carson. You bet. That's the next slide. Um, and they will gain things that they need as adults, like wings, things like that. So the pupal stage is pretty they are pretty vulnerable in that stage sometimes. All right, next. So what are leaf miners or stem miners? Imagine a leaf. I'll hold it up on my, I don't know if you can see me, but imagine a leaf, and this is a house for these leaf miners. And on this house, you might have the top of the leaf. Imagine that that's the ceiling, and the bottom of the leaf is the floor. They are chewing their way through the room. So just as a coal miner hollows out a mountain, these are hollowing out the inside of a leaf. So that's what leaf miners are, and they're doing it in their larval stages, and sometimes they pupate in the mine as well, but sometimes not. And I want to argue that it's a lifestyle. It's not just one taxonomic group. So there are beetles, flies, moths, and wasps that do it in the group sawflies. Wasps in the group sawflies. So to illustrate that point, if you look at the if you look at the figure below, you'll see a leaf with two mines on it. On the right is a mine made by a moth. The larva's feeding under the surface. And then on the left, you'll see a mine made by a fly. Next. So here you have that leaf, and this leaf has damage. You can tell leaf mining damage from other damage if you zoom in. Next. And next. You can see there's a hole in it. That hole is not a leaf mine. It's damaged from the surface, so something sat on the leaf and ate it. Next. But then you have a leaf mine over here made by the moth, and then next leaf mine over here made by a fly. Those are feeding underneath the surface. They're in between the top and the bottom of the leaf. All right. Now, to illustrate specialization, insect specialization. So this plant is winged elm. It's a common plant around us. It's a tree or shrub, and it doesn't have just mollusks feeding on it. It also has leaf miners feeding on it. So, for instance, next. There's a fly species that feeds on it. It feeds on other species of trees as well, like hackberry, but not a huge number. So hackberries and elms are what this fly feeds on. Next. And then there are beetles that eat elm as well, that are leaf miners. But these back. Those beetles are not specialized only on elms, but they do feed on elms. Next. There are also moths on that are leaf miners on elm, and there's one. Go ahead. 2, 3, 4, 5, 6. This is a cocoon and a mine, and then a stem miner. So that's 7 moths that mine elm, and those are mostly all specialists on elm. Okay, next. So I became addicted to these leaf miners when I was looking for a lab exercise to do with my students back when I was a professor 10 years ago, a little more than 10 years ago. And I found this leaf mine on this plant called horse sugar. And I wondered what it was, and no one could tell me. A friend of mine introduced me to Charlie Eisman, who was writing a book on leaf miners of North America. And he took a look at the photos and said, next, and this is a picture of the larva, and said, it's probably a new species. And my jaw dropped. I thought, wow, because I'd always dreamed of finding new species, and I never thought I'd actually do that. And so that was pretty amazing to me. And so I started looking for leaf miners all over the place. Next. So when I think of where we go to discover species, I always thought we'd have to go to the tropics, the rainforests, next. Or the deepest oceans, next. Or a roadside, or next. Or my backyard. And so I found new species in my backyard, which is kind of crazy. All right, so I want to talk about 3 different. Oh yeah, note that these roadsides have been sprayed, so that actually decreases the diversity and allows us not to, and will keep us from finding cool things on the roadside. So I want to talk. I'll talk more about that later. So next. Okay, so I want to talk about 3 different types of discoveries one is range expansion, so new locations for known miners. Number two is new information about known miners, so that could be new food plants that they eat, so host range expansions, or new behaviors or life stage information. And um, and then finding new species. So I'll give examples of each of those next. Oh yeah, and then I want to tell you where to look as well. If you want to find these as well, you know, I've been doing this for 10 years, but I don't have better eyes than any of you, and so you guys could also find these things too if you knew where to look. So one place that I always look is if there's unusual plants, plants that I don't normally see, or number two, common plants, even common plants, plants that I see everywhere. Maybe there are some common plants on which I've found new species. So really anywhere, just look anywhere and you might find stuff. All right, next. All right, so here's the first, so the one example of a range expansion. So this is a fly, Liriomyza schmidti, and it was not found north of Florida till we found it in South Carolina and North Carolina. And it's a generalist, unlike many of the leaf miners. This one is actually a generalist and feeds on a lot of different species and a lot of different plant families. But this is one of them, the Gelsemium sempervirens, and I found it today on Virginia creeper as well. So anyway, so yeah, so it's here and it's around and it feeds on a number of things. Okay. So an example of a host range expansion. So, by the way, there's also a lot of new state records that I've found over the years and that have been found by other people as well. All right, so host range expansion, so new food plants for known miners. In this lawn is next is a plant called Veronica, and if you enlarge that next, you might see that's the plant, and next, if you enlarge that leaf right there, you'll see a mine on it, and that mine next looks like that, and it's a mine of a fly that looks like this. This is actually an introduced species, and it feeds on plants in the genus um, Veronica, and these are next three new host plants for it in the genus Veronica, one of which is a native host plant, which I don't know how much damage it does to the natives, but probably not a huge amount. But at the same time, it does. It's introduced. This is one case of an introduced miner feeding on a native species, among other things, among others. Most miners that I study are native, though. Oh, so Leah asked a question about range expansion and what factors affect range expansion. The problem is these are so little known that we don't really know. I mean, climate change could be a factor, but it could just be that we didn't notice it and that nobody had noticed it before, and it was there all the time. And so these range expansions might be real range expansions, or they could be just our knowledge of their range, an expansion of our knowledge of their range. Yeah, it could be any of those things. It's hard to know for sure. So, oh yeah, go ahead. So next. All right. So there's another one. This is a mine on a sandhill plant called Carphephorus. Next. And it also eats, the same miner was found to eat Biden's bipinnatum. So that's just growing right outside here where I'm sitting. And so it has a broad host range within this family, the daisy family. Next. And it looks like this. It was named for this Carphephorus plant that it eats, but it also may eat a bunch of things. And in fact, it might actually be lumped with another species soon. We'll see. Okay, next. This is another case of not just host range documentation, but also new information about larval stages and about the early life cycle of this species. So this was described as an adult about 100 years ago, and by Braun. And about 100 years later, it hadn't been documented by anybody else. And I found it again and this time by looking at for larvae, and I didn't know what I was finding, but I found a larva mining the Houstonia purpurea, the summer bluet, and it turned out to be this species. So now we know what it eats. And I also found it on a gallium species, a bedstraw species called Gallium uniflorum. So those are the two species that are shown here. And so now we know it's about its early life cycle. Okay, so now about new species. Before I talk about new species, discoveries of new species, I want to talk about how we name new species and what's involved in that. So next, there are sort of three things involved in naming new species. The first one is you have to figure out if the species is really new. If you think it's really new, and why. Next, then you have to convince other people that it's new. Next, then you have to choose a name for it. And I'll talk about each of those three things. So let's talk about the first thing. So to do that, I talked with Owen Lonsdale, who is a collaborator and has helped us describe many of the fly species that we're working with, and or lump them if they need to be lumped. And there are some challenges that he brought up that might affect whether we can describe a new species or not, and how easy that is to do. First one is the group well studied? And if it's a better studied group, that means that we have a better chance of comparing our species to all the known species. Next. The next one is the group diverse? If the group is really diverse, then we have to compare our species to a lot of other species, and that's a lot of work. Maybe back a little bit. So this agromysid flies are the leaf mining flies, and there are more than 800 species in the US. And so there are probably 160 something species in North Carolina. And so it becomes difficult to do this. And if this group is, we're just studying it now and just figuring out more and more species for this group, that means it's not as well studied. So that's two strikes against us. Next. The other thing is we have to find characteristics that tend to vary by species in that family. So we can use external characteristics, meaning what they look like on the outside. We can use their sexual parts, because often sexual parts fit together like lock and key. So we think that if we look at sexual parts, then we'll have some variation that matters by species. And it turns out that males vary more than females in this particular group. That's not always true. So we look at male genitalia in these flies, but we could look at other genitalia in general in other groups. And then we also use DNA. So here's an example. We come up with a sort of a hypothesis about whether our species is new based on whatever evidence we have. And that evidence, if new evidence comes to light that contradicts that, then we might just revise that decision. So next. So for instance, this right here, you don't have to look at this whole thing. This is a hypothesis about the relationships between species. And so that's what this is. It's like a family tree of a group of species. All you have to look at is the red one. The red one is what we thought was one species. It may not actually be one species. This is showing that this quote unquote species occurs in two locations on our family tree. That means it could actually be more than one species. Okay, so that's just an example. So we can use DNA and other characteristics to sort of help us figure out whether these things, whether we have one species or more than one species or whatever. Next. So here's another example. These are two flies right here. Next. And these two flies, we can look at their external morphology, what they look like. And on the outside, and they look fairly similar, but there are some differences. If you look with a microscope, you can find some differences. Next, you can also dissect them and look at their internal parts, their sexual parts. And so their sexual parts differ a decent amount. These are male genitalia for these flies. And they look different from each other. So corresponding parts look different between the two species. And their DNA should tell the same story. All right, so next. So here's a hypothetical example. So let's say we think we have 5 species in a group, and we can tell them apart, so we can distinguish them. We think they're separate species, and we can distinguish them by external morphology, their color, and maybe, but also maybe their sexual parts, and maybe their DNA is different as well. All right, then we discover a whole bunch more species in this group. Next. And some of the characteristics of these new species seem to overlap with those of the species we knew. Oh no, what do? Well, we look for additional characteristics that might help us tell these species apart. And so we have to look for more and more characteristics. And when we do that, we might find that some of our species, that we can tell them apart, and that we now have a bunch of new species in this group. However, if you look at the yellow to red group, it turns out that those new species that we found actually clouded the issue and made it look like we've actually got one species there instead of two, and maybe we do. Maybe that yellow to red thing is actually one species, not two. So it might change what we think of as how many species we have by helping us combine species or helping us separate species. The more information we have, the more we can discern what's true, hopefully, we think. All right. The other point that I'd like to make is that natural history matters, because by looking at the natural history of our organisms, what their early life stages are like, when they come out, what plants they eat, their mining characteristics, all give you different characteristics to use in addition to the external morphology and the sexual parts and the DNA. These are all characteristics that we can use to distinguish among species, potentially. All right. And so we can use these things to describe the species, to say why we think this species is different from other ones and what characteristics it has. So when we say the species has been described, it's been formally sort of described. All the characteristics that it has have been described and put into a document that then gets published. All right, so here's an example of how you can use some of these mind characteristics and other natural history characters to try to distinguish among species, or at least give you a clue that you might have a new species. So this is a real example. This is a mine of a species and a pupa of a species that feeds on wisteria. It comes out during the early part of April, late March, and it feeds in a mine on the leaf, and then it exits and pupates in the leaf litter, and 1 year later the adult comes out and does it all over again. So it spends most of its time hibernating, most of its entire life hibernating, which is kind of crazy. This one also feeds on black locust, and you notice that this mine looks like a blotch. So it just feeds back and forth and back and forth and back and forth and along the leaf and makes a blotch. And sometimes it makes a linear portion at the very end of the mine, but never at the beginning. Next. And this is what it looks like. And all right, and that's what its name is. Next. I found other mines on wisteria that started with a line and ended with a blotch, which is the opposite of this one. The first one starts with a blotch and ends with a line sometimes. This one starts with a line and ends with a blotch. And this one turned out to be a new species, another new species. Can you go back actually? Sorry, I don't understand, Lee. I don't understand the question. An introduction.
Are those
Oh, no,
introduced
these are,
species?
I don't think, good question. Very good question. We have no idea. It does not appear to be a Japanese species. It does not appear to be a Chinese species. We don't know. It's a species that doesn't appear to, we don't know what it is. Neither of these species. So seems to be introductions that we can tell. And they're only known from, well, so Soka, Agromyza soka is only known from black locust and wisteria and Chinese Japanese wisteria. This one, the second one, is only known from Chinese Japanese wisteria. That's it. It does not appear to have a native host that we know of, nor does it seem to have a counterpart that we know of yet in Asia. So I have no idea. It's really weird. So next, go ahead. The good question.
That's wild.
It's wild. It's crazy. Okay, so next thing, how do we name species? Well, species names come in two parts. So they're two words. One is this is the binomial nomenclature developed by Linnaeus, and we have a genus name first and then a specific epithet after that. And so Wisteria, for instance, is Wisteria sinensis. Okay, so species can be named for their external or internal characteristics. So their external parts, their sexual parts. They could be named for their host plant, their location where they're found, or for people we want to honor. So this one in particular, next, is named for sexual parts. Oh yeah, they follow Greek and or Latin rules, usually depending on what the root of the word that you're looking at. So this one is named for sexual parts, crazily enough. Anyway, next. These are all named for the plants that they eat. Arundinaria, Raleigh feeds on wild bamboo, etc., etc. Okay, and the endings are so that they're associated with those plants. Next. You can name something for a place name. The place I used to work was St. Andrews, and this is the only one, this next one was the only one that I had a hand in naming for St. Andrews. Go ahead, next. St. Andrewsensis. This is the only one I've ever had a hand in actually changing the name of a species. For job security at the time, I don't know, whatever. Next. And then you could also name them for people you want to honor. I had nothing to do with this one, but I helped discover it, and I guess they saw fit to name it after me. So this is the only species I have named after me. All right, next. All right, so this is overall results so far, and I'll give you examples as time permits. So there are at least 164 species of leaf mining flies in North Carolina so far, and there are probably more. And at least 18 of those were new to science over the last 10 years. And about eight new state records, at least among those. Next. 225 species of leaf mining moths in North Carolina. There are more. And at least 13 additional species are new to science. There are more of them than that. And then there's more new species in Florida. There are many new state records of these, probably over 100 since 2015. And some of them discovered by me, some other by other people. So other people have been involved in this work as well. So here's an example. This is the wild bamboo, native bamboo, and on it we found mines that look like this. Next. And with larvae inside, and next. And we found that there were 3 new species. 1, 2, next, 3. 3 new species on these things in 2 genera, which is kind of crazy. And that's been kind of cool. So I've been trying to look at other species of Arundinaria, and so far we found one of these species, at least one of these species on another Arundinaria species. So that's kind of cool. All right, next. This one is another example. New species found in Duke Forest. Next. This one was next. You found this one was found in Lee Farm Park, and I found it at Durant and a few other places as well, and on the coastal plain as well. Next. This is now on to moths. This is a moth, new species. I did not discover this one. This one was discovered before I got there, but I helped rear it for the first time. This one makes a long mine and then pupates in a leaf fold at the end of the mine. And it looks like that. Next. And these are 2 species in the genus Buculatrix, one on elm and one on the tiny little dissected leaves of dog fennel, which is pretty crazy, that they can feed on a plant that with leaves that small. Next. Another one, a bunch of new species came from live oak. Live oak's been around, people have looked at live oak forever, but it turns out that people just weren't looking for leaf miners. And so when we started looking for leaf miners, we found that these species are a lot of these species are probably new. Next. Another one on Styrax, which is a cool plant. Next. Yeah, these are some other new ones that at least new information about old ones or new ones. Some of them are, so this is a hodgepodge of examples. So the first one was a new state record. The second one might be a new species only reared in Oklahoma before I raised it in North Carolina. The third one, Lyromyza asclepiadus, is a new state record. The bottom row, Chromastobombesia, probably a new species. And then the second one was not a new species, but we didn't know that for sure that it was a minor. Now we do. Now we know that is a minor when it's young. Next. I still haven't solved this one. This is the first one I started with, and we still haven't raised it to adulthood, and we may never succeed. These marmoras are really hard. They mine from the leaves into the stems, sometimes just the leaves, sometimes they mine into the stems, sometimes they just mine the stems, and they're very hard to rear, but I've managed to rear a few of them, and yeah, anyway, they're really hard. I think I can skip over this part because so I can skip the next couple slides. There are several new species of those. Yeah, you can go faster through this. So this is just the founding cocoons of these marmoras. And oh yeah, now I'll go back. These are a few mysteries. We'll go back one more. These are some new mysteries. One more than that. That haven't been solved. These are all flies in the genus Ophiomya, and these are there's are host plants that they're on. The names are host plants that they're on. These are mysteries. I don't know what they are yet. I've raised adults of the first one, but we don't know whether it's new or whether it's a known species. The other three haven't been, oh yeah, there's another, the ones on the left side have been reared to adulthood, but we don't know if they're new. One of them is probably new. The other one might be, we don't know. Either way, the ones on the right, we haven't raised to adulthood yet. We don't even know what they look like, but we know that they're Ophiomya. The one in the bottom left corner, we didn't even know that it was an Ophiomya for a while. We couldn't tell if it was a moth or a fly or what kind of fly it would be. We didn't know what it was at all for years. And then we finally succeeded last year to raise some to adulthood and figured out what genus it was in. All right, next. This is another one that we've raised a little bit, but we don't know if it's a new species or not, but it might be. Next. Another one raised from Durant Nature Preserve. We've never found this anywhere else, but hopefully we can find it other places. And it's on dogwoods. We don't know what it is. We know what genus it's in, but we don't know. There's none recorded in this genus on this plant. Next. This one we don't even know what it is. We have no idea. I've only found it once. And so if you find it, let me know. It's on Hexastylus. It's pretty cool. It feeds as a minor initially, and then gets out of the mine and feeds externally after that. We don't know what it is. Next. Another one on Brackenfern. Again, we don't know what it is. Next. Yeah, if you're looking for minors, you'll just want to look at different times of year. Next, you'll want to look closely, so you want to have magnification with you. Loops are important, or some magnifier, but dissecting microscopes are fun, but not necessary. You can check different plants. If you photograph both sides of the leaf. Also photograph with light shining through. You might see the critter inside. Next. You can care for them if you want. You can put the leaf in a vial with a little bit of paper towel at the bottom with a little bit of water on the paper towel, just a few drops. Next, and label them. I label them. Sometimes they need soil to burrow into when they're pupating. Usually not, but occasionally they do. Next, I use a paintbrush for moving them around. Next, I preserve leaves in envelopes. I preserve flies, wasps, and beetles and pupae in ethanol, and I preserve moths in gel caps, like empty pill capsules. Next. Then I report them on iNaturalist, and that's an important thing. There's a Leaf Miners of North America project. You could report yours and add it to that project. Next. Sometimes people add to Bug Guide. I used to do that more than I do now. Next, That's another site for identifying bugs. There are threats to these. I want to say that we're spraying our roadsides a lot more these days, and that kills plants, which are the food plants for these things. So miners and other insects may be in danger, and they are. I mean, we know that insects are probably in decline in parts of the world, and in many parts of the world, and this is one of many reasons why. And I think we need to be thinking about the poisons we put into our world and how often we do that. Next, and I see your question, Lee. Um, um, I'd like to thank a lot of people who helped me out with Species ID, access to field sites, and help documenting species. And yeah, and I'll take questions next. And okay, so I see a question from Lee. Great question on Lespedeza cuneata, which is Chinese bush clover. There are 3 or 4 native miners that feed on it and I've raised several of them to adulthood. We don't need to do that. Yeah, we can just go back. Yeah. This is as far as we need to go. So I don't know how well they would control Lespedeza. There doesn't seem to be controlling it much. The problem is they're feeding on a few leaves here and there, or parts of a leaf. And if they do that, they're really not going to be great at controlling them. You'd need large numbers, and sometimes they are in large numbers, but they still don't hurt the plants that much. So there are only a few documented cases of an introduced or a miner on an introduced plant, horse chestnut leaf miner, I think, that with measurable damage on the plants. I don't think, yeah, I don't know how well they would, how good of a candidates they'd be for invasive plant control, but I would guess not very
Yeah,
much.
I
But I
I
don't
was
know.
just curious.
I am
We've
not
got
sure.
the kudzu bug
Yeah,
now that's damaging soy crops in addition to
yeah. It's a
kudzu
difficult problem
infestations,
for
and so
sure,
like
for sure.
it's
Yeah, yeah.
definitely a questionable thing.
The problem is that you have, I mean, I think that they can be really useful. I think insects, control, biocontrol can be really useful and really worthwhile. And there are some real success stories. There are also some real, you know, issues too. I mean, that it caused by certain introductions. But at the same time, yeah, I'm not sure. But I don't know if leaf miners are good candidates for that. I'm not sure. But yeah. Um, all right. Any other questions? I didn't talk about beetle discoveries or softfly discoveries in terms of leaf miners, but from I don't think I've have very many of those, so I don't think I have any of those that I know of. I've found new hosts for some beetles, but I don't know whether I don't think I found any new species of beetles that I know of. Not yet. It's not
Tracy,
clear.
I've got a question. I know
Yes,
you have been working on a website for leaf mining flies
I
for the
have.
biodiversity project. Is that still in the works, or?
It is. We have a species list that is good for now, and we've uploaded some specimens, but we or some some observations, but we need to develop a way to consistently do that. And the basically it's a technical issue, but we're working on it. And once we can transfer sightings from, for instance, iNaturalist, the leaf miners of North America project, to the site. I think if we can do that in bulk, that would be the best thing. But we have to figure out what information we need from it. And so we figured some of that out, but we need to know how that translates into a file that we would then upload. So we're working on that. And then we're going to open it up to the public. That I really want to do that in the next little while. So I'm working on it. species accounts, that's going to take forever, but we'll see. I'm not sure if I'll do that. That's not a priority yet, but I want to get it up to the public so far. So I'm working on it. And we've got 150 something species, 152 species, I think, represented in our list that are, I think that are actually most of which, I think all of which are actually here. There's a few in the list that aren't here, but most of them are.
Are you guys doing this in INAT?
Not in INAT. There is a
Okay.
Leaf Miners of North America project. So if you want to look at the Leaf Miners of North Carolina, that way you can. But so you can go into the Leaf Miners of North America project and use for the species field, you can type in Diptera and which are flies, and in the location field you can type in North Carolina, and then it'll give you some of that information. Unfortunately, it's not really complete. And the new species, of course, you can't give them a name yet, and so therefore they won't be separated out from the unknowns. We're working on a site through the NC Biodiversity Project. If you haven't heard of the NC Biodiversity Project, I would recommend checking it out. There's a website on many taxonomic groups, not all of them, but many of the taxonomic groups, as many as we can get experts for. And there are experts in the state who are working on each one of those websites. And so each website, there's an overarching website, and David George is actually heavily involved with this. And then within that umbrella website, there are a bunch of smaller websites associated with each taxonomic group. So right now I'm working on one for flies. We don't have any for flies at all, of any group of flies. So I'm working on one for the leaf mining flies, which is only one small group of flies, but it is a diverse one. So at least we'll get a few species in for that.
Okay.
And hopefully we'll add more later. Yeah.
Yeah. Thank you for the ways to reach out. I
Yeah,
appreciate that very
great.
much.
Yeah, sure. More questions.
So
Otherwise,
Tracy,
if
you're
we have
saying
time.
if I
Yeah.
find a species myself, I can name it like the Chicago Cubs,
I
whatever.
mean, I don't feel like I
It
have
has
free
been
reign over
done.
naming species. Yeah. Unfortunately, I don't have free reign. Or fortunately, maybe it's fortunate that I don't have free reign over naming species. I only pushed for it in one case. Usually, we want to name species in as practically useful a way as possible, and therefore we often name it for the host plant that it eats, occasionally for the location. But if we've got a whole bunch of species named for a given plant species, maybe then we can vary it up a little bit and try naming things after the cubs or whatever. Yeah.
I
So you're
just
saying
always think
Wrigley
about
Field.
Lucy.
You never know. It's possible. I imagine there must be a species out there named for Wrigley Field. I don't know, but who
Right,
knows?
the ivy they have growing everywhere. Anyway,
Yeah.
Any more
Right.
questions for Tracy?
Thank you so much, Tracy. That was very illuminating and
And this
super
is what I do when
helpful.
I'm at work and not at work. Yeah. Yeah. All right. So thanks a lot, and yeah, I appreciate you guys listening, and if you have
Well,
questions and want to reach out to me, feel free. Dan knows how to get in touch with me. How people can get in touch with me.
And this will be shared.
this will
Yes,
be
I hope so.
shared.
I will
We
share the
wanted to
link as soon as I have it.
Yes,
Perfect.
absolutely. We everyone, we wanted to thank you, Tracy. Again, it was very informative. I learned a ton, which was fantastic. Also, just to remind everyone that's here that you will receive a follow-up email with a link to the recorded presentation. And everyone working on their EE certification, you will also receive an email with a signed Form B. And that ends our meeting for this evening. And we just wanted to thank everyone for being out here. Thank you so much for spending some time with us.
Thank you, Tracy. Thank you, Dan.
Thank you.
Thank you all. Have a great night.
Thank you. Bye.
Thank you.
I am stopping the recording now. Good job,
Great.
Tracy.
Thanks.
And it is